And we have our next speaker is Inês. I think that she's been preparing with a glass of wine. Inês has a background in developing a variety of using and developing open source tools for the analysis of Streptococcus, I'm going to mispronounce some things here, Dysgalactiae genomes. These genomes came from both human and animal. In 2018, Inês began a PhD in bioinformatics in collaboration with the University Medical Center Groningen, focused on metagenomics, validation, and reproducibility, especially relevant when analyzing clinical data. Her work is supervised by Dr. João Carrico and Professor Mario Ramirez from University of Lisbon and Professor John W.A. Rosen from University of Groningen. Please, everyone, click applause and welcome Inês.
Hi, so thank you for the lovely introduction. Yes, I've been preparing myself with something, but let's get this show started. Can you all hear me properly?
Yep.
Thank you. So I know coronavirus is sort of the hot topic at the moment, but let's change that for a little bit because dengue is way cooler. No, really. Okay, so I would like to share first of all with you a little story of how I got drafted into this, big event. And it all started with Lee being very kind and saying, hey, you all should apply to this, which I tried to put Emma on the hot spot. And it completely backfired on me. So Nabil made me do this. Thank you. I'm wearing a bow tie for you. And Emma, you dear, I will get to you eventually. I think I already did like 50%. Anyway, back to the start. Let's keep this thing professional and interesting. So I'm presenting you a work that has been recently published about dengue virus, specifically identifying the genotypes of the dengue virus in amplicon or metagenomic sequencing sample. This work was a collaboration with the University of Groningen, for which Professor John Rossen is here. So a little background. So dengue virus, especially the dengue disease, it's very prevalent in various places of the earth, especially in the center of the tropical areas where the temperature is a little bit higher and the mosquito is endemic. So there is a lot of awareness going on about the cares about the mosquito to try to avoid the transmission of the disease. And it goes across the whole planet. There's also a lot of work in making people try to recognize the symptoms of dengue fever, to get them to go to a hospital and get checked as soon as they think that maybe they have been bitten by the mosquito and they might or might not have a dengue fever disease. So the dengue virus is mostly transmitted by Aedes mosquito. It also transmits other viruses such as Zika, chikungunya. So this mosquito is most of the efforts have been doing in trying to control this population to try to stop the transmission, especially when you try to travel to South America or India. You will have, you go to your doctor appointments and they'll tell you to wear, mosquito repellent. This is mostly to try to avoid these type of diseases, but there are endemic to some regions, and there are people that are getting sick because they live in the places where this mosquito cohabits with the human population. There's also a zoonotic or sylvatic cycle, so it can also infect animals. And there's ones that are more specialized in affecting humans. An interesting thing about the dengue disease is that sequential infection increases the risk of a severe form of the infection, which is we call the dengue hemorrhagic fever, and this can be often fatal. So this is from 2019, July to September. This is data from SCDC. And the thing that I would like to highlight is, okay, so here you have mostly the distribution of cases of dengue throughout several countries. You can obviously see that South America is an area where the mosquito is endemic, the disease is endemic, there's a lot of cases there. India and other Central Asian countries are also a big focus. But if you notice that Spain, here in Europe, and I'm from Portugal, so this is very concerning to me, it's actually colored in gray. So it had a case of dengue. And this was not caused by an import case. So this was actually someone that got the dengue disease in Spain, in Barcelona, to be more specific. So the mosquito is definitely spreading to areas, you know, very related to global warming. So the mosquito is actually spreading to areas where this disease was not actually commonly found. More recent data, although, you know, there's a lot of coronavirus talk going on. This is from January to February. Also, they're colder months. So you can see that Europe is pretty much off the hook. But if you look at the United States, the ECDC reports that there are co-circulating serotypes of the dengue virus. And I'll let you know why this is important in a little bit. So what is the dengue hemorrhagic fever? So the dengue virus itself, it presents in 4 different forms, what we call 4 different serotypes, depending on the capsule. And each serotype can be classified in many genotypes. The infection with one type gives little immune protection against the others. So this is not good. So it means that you're being infected once with the dengue virus from a given serotype will not give you immune protection about getting infected again. Also, there is an effect that is called antibody-dependent enhancement. So this means that the primary infection that you get of the dengue can actually potentiate your second infection, and that is where the hemorrhagic fever cases happen. So the fact that your cells recognize the capsule of the virus, it actually facilitates the entry of the virus into your cells. So it facilitates the entry rather than actually trying to do what our immune systems are supposed to do and fight it off. And this is very serious. So it's really important to know what serotype you have been infected with. In your first case, if you are infected, again, to know if it's the same or a different one, to evaluate the risk of you developing an hemorrhagic fever. So again, there are 4 different serotypes, so classified at 1 to 4, very easy. So serotype 1 goes from genotypes 1 to 5. Serotype 2 goes from genotypes Asian 1, Asian 2, cosmopolitan, American, Asian American, Sylvatic. So this is nomenclature that comes from where they were originally found, although they are trying to change it from the 1 to 5 or 1 to 6 nomenclature. Dengue 3 goes from 1 to 5, and then dengue 4 from 1 to 3, and then Sylvatic. Sylvatic means that it's often it's found in animal rather than in human infections. But these serotypes are circulating, and they often alternate throughout time. So for example, this is an example from Thailand. You can see that the serotype 2, which was the major for a few years, is being replaced by the serotype 1. So this potentiates the appearance of the serious cases of dengue fever, so the hemorrhagic dengue fever, because often people get bitten in 1975. It's a dengue 2 serotype, so it's actually going to facilitate the infection with the dengue 1 serotype. And this is seen throughout all the countries where this infection is endemic. So there's also circulating of different serotypes throughout the years. There is a serotype replacement, if you can call it as that. And talking about genomics, dengue is a single-stranded RNA with a genome size of almost 11 kbs. It's not a big virus at all, and it has a single polyprotein that encodes everything the virus needs. So it has the structural proteins of the capsid, the pre-membrane, the membrane, and the envelope, and then non-structural proteins. You can actually here have a diagram of the autoclaving sites of the genome and how it will replicate inside the cells. But how do the different genotypes model transmission and infection? So this is actually the question that we want to answer. Of course, we are in an era of sequencing is becoming more available, more affordable. Everyone has had this talk, you know, this is squeezing. So doing RNA extraction, PCR amplification, and high throughput sequencing is not an unviable method to study this, this virus, even in countries where the resources are not very high, because the cost of high throughput sequencing is not very high, especially when you're working with RNA extraction. This is an RNA virus. So because it's so small, you can actually sequence a lot of samples in a single Illumina machine. And with that aim of facilitating the discrimination of not only the serotype, but also the genotype level, which is based on homology, we developed this tool, well, this workflow that is called DENIM. So DENIM initially stood for dengue identification through metagenomics. There's many people making jokes saying that DENIM stands for dengue Inês Mendes. It's a fun pun. But that was not the goal. So we actually wanted to empower the use of high throughput sequencing to monitor the dissemination of the disease. Our goal here is to make the detection or the identification of a serotype and genotype of a dengue sample as easy as possible. So we have a few requirements because we will be working with clinical samples and they have a few requirements. So it needs to be ready to use for those that just want to click or run a command, but also allow for customization of parameters. It needs to be scalable, meaning that it needs to run on a laptop as well it runs in a high throughput cluster. It needs to be reproducible. This is extremely important. So the results that I get now need to be the same as the results I'm gonna get next week or next month. And another issue that we were worried about is the confidentiality of the data, especially if you're working with metagenomic samples that come from clinical cases. You're gonna have human DNA in your samples and you need to keep privacy of that human DNA. So we would really prefer a standalone approach to this problem rather than a web service. So also easy to explore and share the results. So this is just a matter of visualization of the results. Things that are intuitive, things that are you can interact with are usually the key. So we found a bunch of tools that is going to help us try to achieve that. So Nextflow, Docker, React, and make it available on GitHub. So we picked all these up and we tried to make something. And that something is DENIM. So DENIM is a Nextflow workflow. So you need Nextflow to run it. And it does several steps. So you can go from your raw data to a complete CDS assembly, to a genotyping and serotyping, to an alignment, and then get a tree. So I can break this down into several steps. So in the blue squares, you're gonna have your quality control steps, which are extremely important. You wanna filter out as much as bad data as you can. Of course, as input, it receives Illumina fast use, both single end and paired end. I'm sorry about the noise. Then we do a step where we recover the dengue sequences, and I'll go into a bit more detail about that in a little bit. And we do an initial coverage estimation. So based on the reads that we have left, we can have sort of an idea what's the coverage. This means that samples that have really, really low coverage or have no dengue data on them are going to stop in the workflow right there. So you're not going to waste resources trying to analyze something that actually has nothing inside. Then we have a two-step assembly and correction step. So we first try a metagenomic assembler, a megahit, very familiar. Oh no, lies. We first start with a traditional assembler. So we start with spades. If spades cannot, because it's so stringent, if spades cannot give us a complete CDS, so a complete coding sequence, we will move on to megahit, so metagenomic assembler, which is a little bit more forgiving, if I can say that. Sometimes if we have a complete CDS, that's awesome. We just need to blast it against our own curated database to assign a zero and a genotype. If we still don't have a complete CDSs, probably due to coverage problems, we do a consensus assembly. And then we also have the zero genotype information. We do an alignment with MUFT with all that. We compute a phylogenetic tree, and then we have a report that you can interact with. So about the steps of the dengue identification. So we compiled 2 databases. The very first one has almost 4,000 dengue genomes, complete dengue genomes, that were retrieved from the virus pathogen database and analysis resource, so VRPR. They have to be complete genome sequences. They have human host, although, I have to say that this slide is not true anymore. It's not only human host, we also included the mosquitoes. And the collection year, we went from 1950 to 2018. At the time, it was the time we were working on this. The most interesting data set, and the one that took the most work, honestly, was the representative sequences for assigning a serotype and a genotype. So we clustered the almost 4,000 genomes together with CDHit, and we selected representative sequences of each serotype and genotype. So we try to encompass the diversity in a way that it makes classification as easy as and quick as possible. So if you want to have an idea, here are the trees for the classification. So the first one is only the envelope region, which is the traditional method, and the second one is the whole genome, which we use in our denim pipeline. And you can see that they're very easily distinguishable through the serotype and genotypes. So the genotypes are identified with different colors. So here you have serotype 1 and serotype 2. And if we go down, you're going to have serotype 3 and 4. If you have questions about this, I'm glad to go through them. So a use case, you want to see this in action. Does this really work? So this was done in collaboration with University Medical Center Groningen, especially with Erle Lizarazu, with at the time was a PhD student of John Rossen. He now graduated with a very good thesis, mostly because one of the chapters I was in it. But he compiled shotgun metagenomics dataset, and this is the set that I'm going to use to you as a test case. We also tried it with amplicon datasets. We tried it with a negative dataset to measure accuracy. But for now, we have 9 plus samples and 13 zero samples from patients from Venezuela that he collected during his, I think it was his bachelor. I'm not quite sure. Also, we have 1 artificial sample that contains the 4 different dengue serotypes. So this is an artificial sample where it just, we spiked in the tube the 4 different serotypes of dengue to see if we can actually retrieve the 4 CDSs and identify the 4 CDSs separately. We also have positive and negative controls, obviously. How to run this? Very easy. Use Nextflow. So Nextflow run an R pipeline. Here I'm using Slurm because I'm running this on a HPC. And this is how I pass my FASTQ files. So I have to admit, I had a Binfie machine to run this on. And on average, it took, 7 minutes to analyze each sample. But again, I had a Binfie machine. So this might change according to the power that you have available, especially the mapping steps are the one that are the most heavy because they rely on bowtie. So here I have embedded a report. So this is built with JavaScript and React. First of all, you can see the in silico typing results. You have for each sample that you analyze the classification, the identity, and the coverage, and also the ID of the closest reference that we have in our 161 database for typing. You can also, of course, you can do this, you can sort it by identity. But here is just a visualization, a quick visualization of the typing. We have the quality control. Also, of course, we have some warnings. For example, no data left after streaming. This is normal, it's a negative control, but this one is not so normal. For example, this one has a very low coverage, this sample 91, 115. So there's actually no dengue sequencing data over there. So the analysis for this sample was terminated halfway through. And the same thing for this one, I need to. Here are the assembly results. So one of the interesting things is that it counts how many ORFs you have. So remember that spike sample that we had? Of course, it has a warning saying that it has a large genome size, and it's true. It's 4 dengue genomes inside a single sample, but we managed to retrieve the 4 different CDSs independently, which we were very happy about. This is more statistics about how much of the sample is actually dengue. So we can say that this one was very low, had very low amount of dengue virus in the sample until we go from, this is our positive control. Of course, it's mostly dengue virus. These are the plots, the contig size distribution. Of course, given that the ORF size is around 8K, we expect everything to be around this area. This is just assembly trash. But this is the most, I think, visual and interesting part. We have the phylogenetic tree. I'm gonna just change this to be easier for us to see. So actually in this tree, we include our samples. We include the closest reference in our typing database. So for example, for this whole cluster, the closest one is this one. It's a serotype 4, genotype 2. And we also have the 4 NCBI dengue virus references. They have one for each serotype, and we included that on the tree. Of course, everything here can be customized. You can actually remove, ask our pipeline not to include the NCBI references, or not to include the closest match in our typing database. But here it makes it really easy to see that the collection that we had and the relationship between the virus that we had in our collection. So we mostly had serotype 4, genotype 2, serotype 1, genotype 5. Here is the NCBI 1, so it's different, it's this genotype 4. Serotype 3, genotype 3. Serotype 2, genotype 2, also known as Asian American. And again, our positive control was serotype 2, genotype 5, also known as Asian 1, so they were a bit different. So what do you need to install Denim? It's very easy. First of all, you need GitHub. And I'm sure all of you are very familiar with GitHub. The next thing you need to do is install Nextflow, because this is an Excel pipeline. It requires Nextflow to run. And another thing you need is Docker, because all the software that is used in this workflow is actually packaged in Docker containers. So you don't actually have to install the dependencies yourself. Nextflow allows you to use the Docker containers that we have defined, and it will fetch them for you. Of course, the next thing you need to do is just clone it and run it. Also, this work, thankfully, has been published very recently in Microbial Genomics. So if you want, you can definitely check the paper out. It has a lot more user cases. It goes a lot more in depth in the works of this workflow. It's available on GitHub, again, as I said, and there is a wiki page with some information, especially how to reproduce the cases that we present in our paper. So the actual data sets that we have, we made them available, and if you want to run them yourself and see how it goes and if it's reproducible with what we showed in the paper, you have instructions there on how to do that. So thank you all for your attention. If you're curious, these slides are available online. Here is the link to them. I would like to especially thank the people that have been with me developing this work, especially Ele. And my advisors, João Carris, John Rosson, and Mario Ramirez, and the people that paid me to do this. So Fundação para Ciência e Tecnologia from Portugal that is actually funding my PhD. And here's all the faces of the lovely people that actually helped me do this. And thank you all for your attention. Now, how do I stop the share?
Thank you very much. We have a few questions, more than a few questions for
Yeah.
you.
Wait, let me take a sip.
Yes, and everyone, give a round of applause. Thank you so much for that. We have a few different questions for you. Scrolling my phone, I'm sorry.
Oh, no problem.
The first question that came through, why would the virus not assemble well with spades? Wouldn't that indicate a problem? Like with an example?
So the thing is, we wanted to make a tool that worked well with metagenomic samples, because, you know, that makes sense. It's a virus. It's easily retrievable from blood and serum samples. But sometimes that's not the case. You're going to have amplicon based data. And that is tricky to maneuver. We saw that we had very different results depending if we use a traditional assembler like spades. Usually is the most accurate, it produces the most accurate assemblies. And we saw that sometimes we could not assemble. And the reason why was kind of tricky. Maybe it was differences in coverage. If you're working with a metagenomic sample, you're not going to have an even coverage throughout. Even if you're working with the amplicon sample, you're not going to have an even coverage of your virus. So we have this two-step or two-pronged approach, as Professor Mario Mendes like to call it, where you use both. If you try with spades and you have your full CDS, you're golden. It goes through the pipeline. But if not, it's going to try it to assemble with megahit. And this doesn't really make much difference in terms of computational time or how long it takes to run an analysis, because assemblers right now are so fast that it really doesn't matter. There are still times where you cannot assemble, and then we do a mapping. But we avoid that because obviously there is a bias toward the reference that we're using. Even though we pick the one that is closest, there's always going to be a bias to the reference.
Mm-hmm. This question, I'm going to ask it out of order just because I think everyone would benefit from this. How do you say Joao's last name?
Carriço. Can you say that with me? Carriço.
Carriço.
Carriço, yes.
Okay, great. Good question.
It was a good question.
Okay. How easy would it be to extend this? Oh, I'm sorry, I'm asking out of order. And I did want to ask this other question first. What tool are you using for the reports?
JavaScript and React.
Okay. It was beautiful.
It was a help. I had help with that. I have to say that it was Diogo Silva that helped me with that. Also, the tree, it's still a canvas, so it's actually, I have to give credit what credit is due. You know, the interactive tree was really nice with the metadata annotated. That's still a canvas, but it's mostly just JavaScript with React on top.
Awesome. Okay. I think we have time for maybe one or two more questions. How easy would it be to extend this to differential diagnosis? For example, malaria. I'm already going to mispronounce more things. I'm sorry, guys. You say chikungunya, Zika, are all similar geographic.
Oh, chikungunya.
Oh, okay. Okay. Are all
So,
similar geographic areas and often symptoms. So could you use to detect it from a metagenomic blood sample?
that is a really good question, and that was originally the aim of this project. So we actually wanted to develop this to co-circulating viruses that infect the Aedes mosquito. So you're going to have Zika, you're going to have chikungunya. The thing is, it's very dependent on the database that you use. And because we built this in the modular fashion, so each component, it's its own Docker container, is very, very modular. We can alter it and adjust it very easily. Sometimes it's just a matter of swapping the database. But the goal of this tool was actually to classify, to give you a serotype and genotype. And that actually, it's not It does not happen to all viruses that it's in the AEDs. So would there be a purpose to do something like this for that? That is debatable. And I'm not really comfortable in commenting that because I'm not a virologist. I'm a bioinformatician.
Good. All right, and I'll ask one last question, and then the rest of the questions we'll go to Discord for. The output is almost 1 gigabyte per sample, it seems. So how does this translate to the aim of running it on a laptop that you mentioned at the start?
Sorry? Can you say that again?
Sure. The output is almost 1 gigabyte per sample, it seems. How does this translate to the aim of running it on a laptop that you mentioned at the start?
So disk space is actually cheap. So that was not really a worry of us. We were more worried about CPUs and memory. And the way that we define that is because the pipeline is modular. Each module has different requirements, and they will only run, and this is mostly handled by Nextflow. This is the workflow manager. So because we rely on a workflow manager, it will actually handle the resources that you have available, and it will only launch the next step of the pipeline when you have those resources available. So this means that the same pipeline can run in a laptop as it runs in a cluster. The difference is in a cluster, obviously you have a lot more resources, so we can do a lot more analysis in parallel than you can in a laptop. But that doesn't mean that you cannot run this on a laptop. And I've run this exact same data set in my laptop when I was testing it. And it took longer, obviously. And it takes a lot of space. So it produces a lot of data. And this is mostly due to the intermediary files that it creates. So you have the BAM files of the mapping steps, you have your assemblies, you have your polishing of your assemblies. So you need to map the reads back to the assembly. This is all files that take a lot of space. Also, Nextflow is known to create a lot of files. So it's going to be a little IO intensive in your machine. But I don't see that it's nothing that a laptop can handle, at least a medium to high-end laptop.
That makes sense. All right, well, I look forward to